So I'm going to try and present today kind of an overview of what we do in the lab. And actually, I have little to do with development and at all. But Rony many years ago and a few others have pointed out that actually the work I do might be relevant, and I will try to put that into some context, and I'll point out where it can be of importance. And so in the lab we work with two models. One is monkeys, and we do electrophysiology in single neurons in monkeys. And we also work with humans, which are a great model for humans, at least some of us think so. And we do imaging, fMRI, and I think one benefit is that we try to combine different similar paradigms across the two species and see and get different levels of information, physiological information. And I'll start by presenting mainly the two. This might be better. Yeah. The two areas that we focus on. One is actually the main one is the amygdala, and the amygdala is ancient, evolutionary, but it is highly evolved in primates, and in somewhat similar way to the prefrontal cortex. And so if you look here and you compare the basolateral complex of the amygdala, which is the major sensory input and processor of the amygdala, compared to the, for example, to the central nucleus, which is the output production of a defensive mechanism or other emotionally related survival mechanism. So if you compare this to a long evolution, which is not the correct term per se, but you understand what I mean. So that's in a rat, that's in a cat, that's in a monkey, and that's obviously in a human. And you see how the BLA comparably evolves. And so the amygdala, the main processor of the amygdala, actually evolves a lot, and it actually evolves in correlation with the prefrontal cortex that, as we all know, is so highly evolved in primates and definitely in humans. Also, the amygdala and the prefrontal cortex, and especially the regions I'm going to talk about, which is the medial prefrontal cortex. I'm not going to touch at all on the dorsolateral prefrontal cortex. Many interesting roles, but I'm going to touch mainly on the medial prefrontal cortex and more so on the cingulate cortex. So the cingulate cortex and the medial prefrontal cortex is directly connected. I mean obviously one synapse. So there's a one synapse bidirectional pathway between dense connectivity between the amygdala and the cingulate cortex, which means important things for us. And this balance basically almost any, and I'm generalizing, but any kind of neuropsychiatric condition you would choose to focus on shows some abnormal activity in this pathway. There are obviously other regions that show abnormalities. I'm not trying to say this is the whole story, but amygdala cingulate and amygdala prefrontal in general is a main hub of that is probably disrupted in psychopathologies. And the connection that Rony pointed out years ago, and I noticed in several things, and I didn't know that when I started many years ago, the work, but now I'm more into it, is that actually BLA cingulate and BLA prefrontal cortex actually mature during adolescence and complete maturation only very late in adolescence. And so basically, everything I'm going to show you, and I'm going to show you several models that we do in the lab for understanding mainly post-traumatic stress disorder and/or anxiety, learning models that can lead to that. I'm going to show you how they are related to amygdala-cingulate interactions. At the end, the question, at the end of each project, we can do it like a mantra. At the end of each project, someone from the audience can get up and say, but how is this affected when all what you're telling us happens in adulthood before full maturation of amygdala prefrontal synaptic connectivity. And that, for me, now at this point, is the open stage, where all these models that are highly connected to psychopathologies and relate to behavior are driven by amygdala prefrontal cingulate connectivity. What happens when these learning models occur prior to full maturation of synaptic activity, and hence the balance that in adults can normalize behavior by normalizing emotional, rational, statistical understanding, et cetera, of the world does not exist, and how does it then remains until adulthood. And I think it's not an anecdote, but those, I'm sure there are better, more clinical psychologists and psychiatrists in the audience than me. A lot of those psychopathologies arise during adolescence and only and take some path to end up being this or that or another classified specific psychiatric condition. So that's going to be kind of the main framework. And another framework which I like a lot of using in the lab is actually using what we're going to call normal evolutionary driven behaviors that are all normal evolutionary behaviors when you think about threats and survival, but under slightly extreme conditions can actually result in maladaptive responses that are then exhibited in anxiety, post-trauma, and maybe even depression. So all the behaviors, if you think about it for a second, that I'll show you, are completely normal behaviors unless they are taken to the extreme, and that's their PTSD or anxiety disorder kind of conditions. So I'm going to focus on several behavioral models that some of them we developed, some of them are obviously borrowed from a lot of ideas that many other labs in the field work on. One is the resistance to extinction. Once an emotional memory is formed, how much does it resist to modulation later on, and flexibility of learning that it's not so bad as it should be. Spontaneous recovery, I'll explain later. The other model is that of overgeneralization. How much do you overgeneralize an emotional memory? The other is exploration, exploitation trade-off. How much do you stay in a relatively good place, but maybe you can get to a better place if you explore enough with all the risks that are involved in that. And the final one is that of repetitive insults. Is there any reason to assume that repetitive insults are worse? I mean, they're obviously worse, but under what parameters, what conditions, and what are the underlying mechanisms? So all these, I think I can claim pretty reasonably, are evolutionary reasonable. You want to have a resistance to extinction of a highly emotional, threatening memory because you want to remember that, whatever, a lion just ate your zebra friend. You don't want to forget it. But when you don't forget such emotional memories for us as humans, it actually can be a PTSD trait. Similarly, for overgeneralization, you want to remember that similar things have a higher probability of being dangerous just because they are similar in stimuli. Something that looks like a lion have a high probability of being a lion, but you don't want to overgeneralize and seeing a cat and still behave like it's a lion and run away. So overgeneralization is also a trait of anxiety and PTSD. Exploration, exploitation is actually kind of a new domain. We're not the only ones, but there's been a lot of interest in that. If you're over-anxious, you over-explore, jump too much, and do not settle down in a local place, local normal place, not the best, but reasonable enough place. I'll show you evidence for that. And the final one is actually how, which again makes sense, repetitive insults should actually increase your chance of being afraid of that incident. But again, when it happens too much, it could result in PTSD or anxiety. So all are very normal evolutionary behaviors, as Michel Foucault said in his nice book many years ago, just a matter of where you put the criteria and when it's too far away, it becomes a debilitating disorder. So, okay, so I'm going to give examples to all these four models, and short example and how we investigated them, some in monkeys, some in humans, some in both. I'll start with some older studies and about memories that resist extinction. Some of you already heard that, and I'll just briefly mention a paradigm we developed in the lab many years ago when I started my lab at Weizmann. And we were thinking about how do we do emotional learning in primates, which is not trivial in monkeys. There are many issues to consider. And we kind of figure out that one good way to do it is by using tone odor conditioning. So the benefits of that is that odors, we know the anatomical route into the amygdala. This is a big advantage for us, whereas in many other kind of reinforcers, it's actually very hard, and there are many synaptic unknown ways into the amygdala even today. In the case of olfaction, we know exactly how it gets into the amygdala. We know how tones get into the amygdala from the work of Weinberger, Joe Ledoux, and many others. And another good thing, which is, again, highly important for us, I mean, at least for me and everyone who measures brain activity, is that we can measure the same modality, but opposite valence. So we can use both pleasant appetitive odors and aversive odors, and it's the same modality, but different valence. This is not trivial because a lot of the studies in the field are trying to compare good and bad, and its interpretation is just a little bit harder because they use different modalities. I mean, we do this as well in other studies, so I'm not trying to say anything bad about these studies. I'm just saying this is one benefit of using olfaction. And another nice thing is that we have an implicit measure across species, and the measure is very simple. You see it here, an animal or a monkey simply reduce it. You see the red dot over there? It's pretty hard to see, but okay. So an animal or a monkey or a human simply reduce or increase the spontaneous breathing as a response to an appetitive or an aversive odor. And we can also measure learning, which is the preparatory response to the tone, to the odor that precedes. So that's being learned. The tone predicts the odor, and we have learning for that odor. So that's the model that we're kind of going to use in different manipulations across most of the talk. Some of the studies are not using this exact paradigm, but that's the basic for a lot of it. And the nice thing, again, is that what I like is that we can use the same model across species. And when I say species, I mean, in my case, monkeys and humans that do the exact same paradigm and then do fMRI and electrophysiology and see different levels of explanation. Okay, so the first has to do, as I said, with resistance to extinction. And Uri Livnei in the lab many years ago had a very nice hypothesis, and he asked what can differentiate persistent memories that are resistant to extinction from memories that are easily extinguishable. And going back to 1960 psychology, which is usually, that's usually what I do with students, send them to read all of the psychology literature in the 50s, 60s, and 70s. That gives the best background to any student. But in this case, it was really worth it. And there was a paradigm that, partial, which some of you might know in the psychology, partial reinforcement extinction effect. I'll just describe it here briefly. If you condition, if you condition an animal, the pointer is here, okay. So if you condition an animal, each pointer is 10 trials. If you condition an animal, but in one day you condition it with a one-to-one relationship, every CS, every tone is followed by an order, you get nice learning, and then you get immediate extinction. So extinction is when you present only the tone without the order. So the animal immediately stops to respond to the tone because it doesn't predict an order anymore. However, if you do 2 to 3 relationships, stochastic relationship, and some uncertainty injected into the paradigm, some of them predict the US, some of them don't predict the US, and it's obviously randomly interleaved, then the animal, surprisingly or not, does not distinguish, and not distinguishing does not extinguish, and in this case it means over 30 trials, some of the animals don't extinguish at all after 30, 40, 50 trials. Some extinguish after 20, 30, but in any case, it's highly different than the immediate extinction you see. And that brings a very important, a very nice controlled study, because if you look here, you have a controlled state where the memory looks equivalent, but it's going to be extinguished faster afterwards, whereas not extinguished afterwards. Although, again, the manipulation here is exactly the same. So the only conclusion can be that the memory here is stored by some other mechanism. And so that's what we find. We recorded in the amygdala and the cingulate cortex. And I'll give just, I gave a motivation, but amygdala, I think I don't have to give much motivation. What has to do with emotional learning. The cingulate cortex first has a lot to do with in anxiety patients, always lights up in imaging studies of anxiety patients, but on the other hand has a lot to do in statistical learning of understanding the environment. So it's a good hypothesis why it would fit in this kind of learning. And so if we do that, that's exactly what we find, and what I'm showing you here are just neuron to neuron, single neuron correlations, one neuron from the amygdala, one neuron from the cingulate cortex, average over many, but these are two-dimensional correlations. And what you can see is summarized here, and that is that in the case of the one-to-one relationship, you have immediate correlations going up, synchrony between the amygdala and the cingulate, going up and then dropping down until the end of learning. And in the partial relationship, it's the other way around. You see those correlations kind of grow up and stay, even though, I remind you, here, the memory was exactly different. Now, if you now try to correlate the two, and I'm taking physiology from the end of learning, so physiology is taken from the end of acquisition, which is right here, and I'm taking behavior from the end of extinction, how much they could extinguish, then you see a very nice correlation, meaning the more there was synchronous activity between the amygdala and the cingulate cortex, the harder it would be for the animal to extinguish this memory. Now, I'm not going to go into the whole story of why it makes sense. There are also interactions with the ventromedial prefrontal cortex that is responsible for extinction, and that fits with that model. But the basic understanding is that when the amygdala and cingulate are combinedly involved and in a synchronous manner, the memory is more stable and harder to extinguish. So we went on and trying to do, we have a big part of the lab is computational and trying to understand reinforcement learning mechanisms, and we tried to see what kind of error signals are actually propagating in the network. And to cut a long story short, classical reinforcement learning theory has two kinds of errors, which you're probably familiar with, the Rescola-Wagner models and similar models. And one of these errors is what's called the absolute error, kind of like attention. That just means something has changed. The other is a signed error, which means it changed for good or changed for worse. And we can differentiate these signals in the brain using a slightly more sophisticated reversal paradigm. And what we find is that, and I'm going to emphasize what's important here, is that we can measure at a millisecond resolution the transition of the signals between neurons in the amygdala and neurons in the prefrontal cortex. And what you see is that the condition for flexible learning, for being able to move from safety to switch contingencies and move from safety to danger predictions, determine there are two conditions that are necessary for it. One is that you need an attention-like signal coming from the amygdala to the cingulate cortex, and the other that you need the sign cortex from the amygdala. So, I mean, in simple words, it's like the amygdala saying, look up something has tremendously changed, and the cingulate cortex collects information from other regions, likely the striatum, the dopaminergic system, etc., and says, okay, now I'm going to tell you what exactly have changed, and you can correct it in the right direction. Okay, so that's kind of trying to understand what makes a memory flexible or resistant to extinction. But there's a big problem in the clinic, especially, but one that is easy to replicate in the lab, actually, and that is that if you extinguish a memory, I'm not going to go over all conditions, but if you extinguish a memory and then simply wait for some time, then the memory simply reappears. And that's called spontaneous recovery, and it's a real problem in the clinic. People undergo exposure therapy, which is driven by extinction IDs, and then they just come back sometimes weeks, sometimes months, sometimes even years later to the clinic and have the PTSD symptoms again. So there's spontaneous recovery. So based on that, and based on what we saw before, that the cingulate cortex is required to maintain persistent memories together with the amygdala, Oded Klavir in the lab, postdoc now has a lab in Haifa, has hypothesized that if we can do deep brain stimulation, which is something we're now collaborating in clinical trials as well, can do deep brain stimulation in the cingulate cortex and reduce its activity during extinction, we might be able to prevent the spontaneous recovery. And so that's exactly what they did. We put an electrode in the cingulate cortex. We use low-frequency stimulation for many reasons I will not discuss now. It has been shown to induce LTD in many ways. You can do it with TMS, by the way, as well. And what we do is we do low-frequency stimulation of 1 hertz during the extinction. And you see here the 2 lines of extinction. And I'm not showing you learning because it was the same. So one day it was with, the behavior is the same. We just show the CS only. And one day is with deep brain stimulation, the other was with not. To our surprise, there was nothing in the extinction itself. But if you look 24 hours later, what you see is amazing in my view. So this is kind of surprising because it's the other of what people are used to look at. This is actually the normal result. Animals and humans almost always have a spontaneous recovery on the day after. So they go back to responding to the CS. And so that's what we saw when we did not stimulate, When we did stimulate, you see no spontaneous recovery at all. And so that's 24 hours. We also replicated this for 36 hours to show that it's robust over longer timescales. Why 36 hours? Good, because reviewer asked us to show that it's for a longer time. I didn't want to repeat this as a monkey study, which is crazy to repeat, but then I remembered that students are lazy and don't work on weekends. So we had natural opportunity to go back and from Thursday to Sunday and check several data points and see that it actually holds for 36 hours, sorry, even more as well. Yeah, so it works even for longer time scales. Here we just show that we indeed reduce excitability, and if we now correlate, this is a modest correlation, it's not perfect, but I still think it's nice, because it's actually a very hard one to demonstrate. It takes physiology from the end of extinction, so how much we were able to reduce excitability in the cingulate cortex, and it correlates it with the next day, 24 hours later, amount of spontaneous recovery. So physiology for one day, behavior from the next day. And you see a modest but reasonable correlation of this. Okay, as I said, in extracellular recording in monkeys, it's very hard to know if it's really LTD or some other mechanism. And another problem is when we stimulate in the cingulate, it's really hard to know, do we affect really amygdala to cingulate, or do we affect any other pathway? That's electrical stimulation. There are many pros, but many cons. So we repeated this with Ofer Izar, a wonderful optogenetic expert at Weizmann. Again, Oded pioneered the study, and what we did was injected in the amygdala, but this time shine the light, the laser, only on the cingulate, in this case actually in the prelimbic in mice, and we could then identify specifically that it's a result of amygdala to of BLA to prelimbic synapses. I'm just showing you here that we can induce long-term depression. This is normal. This is after the stimulation, and this is the recovery time it takes as different amounts of light of maximum laser intensity was used. So it takes a good 30 minutes to an hour till recovery, and we can show that it actually induced both faster extinction and less spontaneous recovery. So it seems, and that I'm not going to say, I don't think it's completely against the dogma, because there has been several studies before that have shown, but usually it is considered to be prefrontal modulation of the amygdala. And this and the previous studies, I think, are nice by showing that it's actually, as we can all imagine, a much more complex circuit, and the balance between it is what important. So just as prefrontal to amygdala is important, so is amygdala to prefrontal, and the balance between the two pathways actually determines the rate of, in this case, of learning fearful or threat kind of memories. Okay. Finally, we wanted to go back to the primate and still show that we can show that in the primate there's the same directionality of amygdala to prefrontal, to cingulate cortex. I mean, I was convinced by the rat studies, but still, before we go into real studies in patients, in human patients, I wanted to replicate this in the primate. And so optogenetic in the primate, we play with it a little bit, but it's still extremely hard. What we could do is look at local field potentials that reflect the synaptic input. And what you can do then is look at local field potentials recorded in one area and average them on spikes, the output mechanism, on another area. And if you do that, what you find is, again, is that amygdala spikes are the other way around. Cingulate cortex, theta activity, local field potential, the theta range, which I hope, I mean, it's clear to everyone why this was one of our hypotheses. There could be other bands, but theta is definitely other bands, and we tested all other as well. So theta oscillations in the cingulate cortex were locked to amygdala spikes, but not vice versa. And it was also highly appropriate, highly suitable with the learning process. So this was rising and then falling as this is behavior, the blue one. And once behavior is complete, there's no more synchrony needed between the amygdala and the cingulate in this specific case, and this validates that these points are similar across trials and animals. So we see that in the monkey primate as well, there's amygdala to cingulate cortex process occurring during emotional learning and stability of memories. Okay, I'm going to jump to the next model, a model that's close to my heart, but I'm going to go over it pretty fast. Again, several labs around the world are thinking about this as a model for anxiety and post-trauma. The idea is very simple: memories that overgeneralize, so it's better safe than sorry, and the intuition, I think, is clear here. But we had the hypothesis that, I would say a bit stronger hypothesis, that this is actually not a decision-making active process, but it's actually a perceptual based process in the following sense, that it's not that people choose to respond similarly to slightly similar stimuli. They actually cannot help it, and they do not discriminate between the two stimuli after aversive fear conditioning. So we did a very classical, simple psychophysical study, which was published already 7 years ago. What we did was simply did a classical J&D 2AFC paradigm from psychophysics, where we measure the just noticeable difference around two tones. Then one of the tones is conditioned with an aversive odor. Again, we can do the same paradigms. And what we see is that, so you can see here, on the upper right, the actual psychophysical functions, psychometric functions, sorry, and you can see that the threshold has shifted. So the intuition is simple. If you can discriminate 100 hertz from 104 hertz before conditioning, that would be approximately the normal range. After the 100 hertz is conditioned with a highly aversive odor, you can discriminate it only from 110 hertz. And it's not your choice. We did many controls for that. You simply cannot discriminate between them. We played with it and replicated it in several studies, different contexts, many things, many controls. You simply cannot discriminate between them. That shows it over the population. So this is the change in J&D and all kind of controls. We did positive conditioning as well, obviously. It was very robust. Another thing that was robust is that it stays for 24 hours, so it's real perceptual learning and not just some short-term adaptation. It was robust to two things, which for me are highly important. So it's replicated now in several. It was robust to using different modalities of reinforcers: odors, aversive sounds, and aversive pictures. So that's one thing. The other is that it's also, that's a collaboration with Galia Vidan at Ben Gurion, is that it's also robust to CS modalities. So we can use basic auditory features, or we can use basic visual features, such as orientation and acuity, etc. They all had the same trait, that the J&D, the discrimination, increases after it's conditioned with an aversive odor. Even more surprising, we could show that it's related, that can even happen after we use monetary losses, which is interesting because it's a secondary order conditioning. We weren't sure that it's going to work, but it works. I'm not a neuroeconomics, so I don't want to interpret it too much, but I think it's interesting and definitely also allows us to use it in the lab in many ways. So that's another way to plot it. It's just a generalization function. How much you identify this is what you were conditioned to, and how much you identify other stimuli as the conditioned stimuli. And so you make many more mistakes when this was a condition to an aversive odor. And when we did this with imaging, we found only one region that was inversely correlated in activity to the behavioral function in purple, and that was the amygdala. So the interpretation here is a little bit tricky. There are two ways to interpret this. I'll give you one which I think is the correct one, but we can discuss it later. So we know that there's a system in the brain that can differentiate 10%. That's above our perceptual acuity for sure in all people. Unless you're hearing damage, and we check that. But if the amygdala sends a signal and the amygdala projects directly to both auditory primary cortex and primary visual cortex, V1, V2, the amygdala projects directly to all in primates. If the amygdala says this is a threatening signal, it kind of overrides. And your response is, again, unconscious. Your response is that you cannot discriminate between them. It's not a cognitive choice, not a decision-making. You cannot discriminate the two stimuli. Okay, we repeated that in patients, in anxiety patients, as the hypothesis goes. And according to the hypothesis, we found nicely that anxiety patients have a wider generalization curve, so they would associate farther away stimuli as the original conditioned stimuli. And this was surprisingly, or not at this stage, this was highly related to activity in the cingulate cortex and in the amygdala. And again, this is kind of a prediction study because what we take is activity in the amygdala and the cingulate cortex during conditioning, but the behavioral measure is the width of generalization in the next phase. So there's a conditioning phase and a generalization phase. So it's activity taken from the conditioning, generalization width, how much you generalize, the width of the slopes from the half an hour later. And you can see here across subjects the prediction correlation. So that was nice. And we can also show that during the generalization, if you now look at MVPA, do pattern-related analysis, and see how much the activity in each of these regions discriminate between the stimuli, then the amygdala is the one that has the highest correlation with the ability or inability to discriminate different stimuli. Interestingly, the auditory cortex also showed the difference, not across valence, positive or negative, but between patients and normals, potentially indicating there's a little bit closing the circle logically, but potentially indicating that in patients, the auditory cortex is also involved. Okay, so we did that in monkeys. We went back to monkeys, recorded in the amygdala, and showed that we can actually find single cell changes. I'm not going to go too much into this, but single cell changes in tuning curve that explain. Now, I'm just going to say one thing here that is important. This is a big challenge for neural network architecture for the following reason. And that confuses many people at the start when we send this to publications of the, well, you should have better distinction. Why do you have worse discrimination on something dangerous? So here's the confusion. That's a delicate but important point. For dangerous stimuli, you want to have better detection, but you want to have worse discrimination. I want to identify a lion in the grass. I don't care which lion is it, and I'm willing to take the risk it's not a lion and overgeneralize, right? Now that makes sense, but it's a real challenge. We did that in modeling. I won't go into it. It's a real challenge for a neural network. How do you balance enhanced detection, which means more activity to the stimulus, but also less discrimination, which means more activity to more stimulus around. So I'm going to go into it, but this kind of solution network solves it in some way. Okay, so repetitive insults, these are relatively new projects, so I'm going to go over them quickly. So I was interested for a long time, how does the brain maintain timescales for more than 2, 3, 4 seconds that we used to do, at least in electrophysiology neuroscience. We look at responses, even working memory, are usually 2, 3, 4 seconds, not much more. But here's something interesting. So if you do the same kind of conditioning, same kind of learning, and now the inter-trial interval in this case is very long. It's approximately 30 to 60 seconds. That's a lot for usually neuroscience studies. You get a trial, conditioning, and then you wait a full minute, and then another trial. This is in monkeys. And what's interesting, and again, this has been described a lot in the classical learning cognitive community, but hasn't been revisited in neuroscience, is that the longer the time between the insults, the trials, the better will be the learning. This sounds maybe weird at first, but there are several explanations for it, interpretations, which I will not go into all of them. It's a behavioral fact, and what you can see here is exactly this. If I now take all the trials and divide them according to the length of the time between the trials, then you can see that the longer the time between the trials, and again, this is between 20 and 60 seconds, then the longer will be, the higher will be the behavioral response afterwards. And even more so, if I do this and look at the learning rate, the behavioral response minus the previous, which is basically the instantaneous slope, you also get a better slope. So you learn faster and better if you had those well-timed. So that's like me, apologize for that, but maybe I should give a combat-like example. If there are many bad things happening at once, that's maybe highly traumatic, but it possibly much less has the chance of creating a traumatic experience than having something. A minute later, another something. A minute later, another something. Maybe it's because the brain lumps all the close by at once, maybe, we're not sure yet. But that's empirical behavioral evidence, and it works very nice in the lab and across species. This is the first time it shows in monkeys and aversive conditioning. It was shown in rewards in pigeons in the classical studies before. So, I mean, but that raises a real puzzle for a neurophysiologist, because we know neurons can maintain time for 2, 3, 4 seconds by persistent activity. We have no clue as a community how neurons can maintain time for 60 seconds. This is much beyond any time scales and constants we know in neurons, synapses, etc., etc. They're not even good theoretical models how we do it. So we went again to the amygdala and cingulate cortex, and I'm going to tell you very briefly, we found neurons that are active all along the long 45, 50 seconds. They have a preferred time during this 60 seconds. And if we look at, again, correlations between the amygdala and the cingulate cortex, we find that they transfer information between them early in the middle, and at the end. So they use, one network uses the other to transfer temporal information and say, I'm now early on in the time, I'm now in the middle, I'm now in the late, and together they bring, and this is using all the neurons, together they allow, I'm just going to focus on this here, together they allow a very good prediction. This is a decoding, standard decoding model, so this is real time, this is the decoder time. I can decode from the neural activity very reliably where am I in the middle along the long time scale of up to 60 seconds. And maybe reassuring is that indeed, if we now look for a population of neurons, we find a population of neurons that starts responding right before the next trial, 60 seconds after. This is, if I let you approximate 60 seconds, you're going to have a huge variability. You psychologists know it better than I do. You're going to have huge variability in estimating this time scale of 60 seconds. We have neurons that actually start responding just before. Okay, finally, I'm not going to talk about that. This is not a method. We find how memories are rehearsed in the amygdala and cingulate cortex by looking at triplets of neurons, and we see rehearsal of the previous memory. We hope it's going to be accepted soon. And okay, I'm going to skip exploration and exploitation, I guess, for the lack of time. We're highly interested in that. I think you can understand the intuition. And we find very nicely that with all kind of models and behavior in humans, that trait anxiety actually correlates very nicely with how much you explore the environment, at least in these well-controlled tasks. We can model it nicely and differentiate the parameters. And again, we see that the cingulate cortex is highly correlated with the exploration parameter. We're still working on it. How does exactly anxiety affect exploration and vice versa, and what does it mean for the learning? But it obviously, the amount of exploration can be useful for learning, but only up to some extent, where you start to jitter between local minima and don't converge into the right solution. So the plus and minus, as always, there's a good place to be, and we're now focusing on that. Finally, I'm just going to give you a promo for a study which I hope is going to be expected soon. I'm not going to say much about it because it's a long, crazy study. It's in collaboration with Yitzhak Fried, and so we exploited the opportunity of recording neurons from both amygdala, cingulate cortex in humans and in monkeys, which is opportunistic, you may say, but it's something I've been wanting to do for a long time. And we find, in my view, a beautiful result that I'm overinterpreted because I'm so excited about it, is that if you look at the order, then you see the human, where is it now? The human cingulate cortex. So we quantify efficiency using entropy rates and information theory measures on spike trains. Oh, sorry. And we also quantify robustness, on the other hand, by quantifying how much neurons are acting together as a population. So it's synchrony and several other measures, again, coming from information theory, measuring how much they use shared vocabulary. And what we find, which is an empirical finding, is that there's a very interesting trade-off between efficiency and robustness. And the human cortex is more efficient and less robust. The human amygdala is slightly less efficient, slightly more robust. The monkey cortex is slightly less efficient, slightly more robust. And the monkey amygdala is the most less efficient and the most robust. Now, if you think about it for a second, it fits both levels of going from non-human to human primates. There is a big evolutionary pressure on cognition and being efficient, and at the expense of robustness, which makes you vulnerable, unfortunately, to the kind of psychopathologies we have been discussing. If you're a monkey or in the amygdala, yeah. You have also signs for minus? Okay, that's a slide, I promise. And if you're a monkey or in the amygdala, you want to be more robust. Well, you want to be more robust if you're a monkey, but in the amygdala, you want to be more robust because you want to produce reliable survival responses, and you care less about the details and richness of the stimulus. And that has been shown in several studies, that emotional memories are less detailed and better remembered. And so I really like this trade-off. And the study is very complex, so I won't describe it at this point, but I think it's, in general, I think cross-species differences are highly important to investigate, and we heard many good talks. Almost all of them were concentrated on this. I think it's highly important. Okay, so conclusions. Multiple normal to maladaptive learning strategies can lead to psychopathologies. On the extreme case, they're all evolutionarily reasonable, but on the extreme, they can be dangerous. We talked about resistance to extinction, overgeneralization, its perceptual origins, exploration, exploitation, they didn't have much time, repetitive recurrent insults. All of them are coded and maintained by a delicate balance between the amygdala and the cingulate cortex. And as Ruti and others have told me, amygdala ACC synapses mature only later, and so it raises intriguing questions, in my view, which I hope collaborations will emerge. What does it mean when all these happen early, before those synapses mature fully? And I'm just traveling, enjoying myself, and these people actually do the work. So many of them have Uri, Oded, Jennifer, Ophir, where is Yoav, Liran, Tamar, Yossi, obviously, and Arya, Raviv. Thanks. Right. Okay. So so we find earlier. You go to the small world network hypothesis, which I think most people will now agree this is right about the cortex. And what you're suggesting is that, which is true, we recorded locally in a small network in the cortex. But if we now expand it to look at other cortices that are relevant, say, insulin for social, we might find that they know how to collaborate in a more meaningful way, just like single neurons in the amygdala are doing that locally because they want to produce a fight-or-flight response. That's a great hypothesis. Right, yeah.